Good morning po, Doc. Yes po.
Naririnig naman. Sige. Since it's already 9:30, let's start na. Can you hear me?
Yes po, Doc.
Yes po, Doc.
Yes po, Doc.
Okay. Sige. So, good morning, everyone. Sorry, we had a delay in our schedule. But anyway, sige. So, we'll finish this chapter this morning so that we will be back on track and we can follow na our regular schedule. Okay. So for this morning, we will be talking about the catabolism of the carbon skeletons of amino acids. So if you can recall, during our very first meeting, diba, we talked about the biosynthesis of the non-essential amino acids, and that was followed by the catabolism of proteins. So beginning with the amino group. So just remember that in catabolism of proteins, so initially they are separated, or they are divided or broken down into the different amino acids. And for the amino acid, it usually starts with the amino group, except for there are 3 amino acids which we will learn later on that do not participate in in transamination, or they do not follow the usual pathway for metabolism. So after getting the amino group in the form of ammonia, and which enters the urea cycle, so the remaining carbon skeleton, or the backbone of the amino acid, also undergoes catabolism. And if you just memorize by heart the on how the non-essential amino acid is formed from the metabolites of the Krebs cycle or from the glycolysis, this will be easy for you na lang kasi it's just a parang the opposite naman. So it's yung pag-breakdown. So from the amino acid going back to alpha-ketoglutarate or pyruvate or oxaloacetate. So it's the opposite of the biosynthesis. Okay, so we have no choice sa biochem. We really need to memorize these things, diba? And also as well as the enzymes involved in the breakdown or in the biosynthesis. So it's really part in our the way on how we study for biochemistry. Okay, so moving on. So most disorders of amino acid catabolism are rare. So actually, they are rare, but if left untreated, they can result in irreversible brain damage and even early mortality. So it is important that we can detect this amino acid disorders during the time of prenatal pa lang or early postnatal, so that we can give timely treatment for these disorders. And usually, it's easy naman to treat this kasi usually they are just diet modification lang. So all you have to do is know the defect and just modify the diet and give low amino acids and give a low and in the diet advise your patient to reduce this amino acid that has impaired catabolism. Okay, so the mutations for involved in catabolism are either of a gene or of associated regulatory regions of DNA that can result in the failure of synthesis of the enzyme or in synthesis of a partially or completely non-functional enzyme. So if later on we will see that in catabolism of the carbon skeleton, there are really many enzymes involved for it to be broken down again and for the amino acid to return back to the Krebs cycle or to the glycolysis. So any failure in those enzymes or any non-functioning enzyme can lead to metabolic disorders of this particular amino acid. So some mutations do not actually adversely affect the enzyme activity, but the mutations that compromise an enzyme's three-dimensional structure or that disrupt catalytic or regulatory sites of an enzyme can have severe metabolic consequences. So the low catalytic efficiency of a mutant enzyme can result from impaired positioning of residues involved in catalysis or in binding a substrate, coenzyme, or metal ion. So very specific kasi ang enzyme that breaks this amino acid. So any impairment lang in the binding site can lead for this enzyme, can lead these enzymes to being non-functional. So and also mutations may impair the ability of certain enzymes to respond appropriately to the signals that modulate their activity by altering an enzyme's affinity for an allosteric regulator of activity. Since different mutations can have similar effect on any of the above factors, various mutations may give rise to same clinical signs and symptoms. So, ayun, for example, a particular patient may present similarly with these signs and symptoms. Actually, if you go down deeper sa molecular level, it will be found out that they may differ in the particular enzyme wherein there is a problem, although they may present with similar mutations. So it is important to determine or to know what particular enzyme has a deficiency or has a defect. Okay, so based on nutritional studies that was conducted in the past, it was established that the carbon atoms can be are interconvertible actually. So from fats, carbohydrate, and protein, and you will see that later that from amino acid they really go back to the they participate in the carbohydrate synthesis and also in protein synthesis. So actually, in our body, these carbon atoms can transform or can transfer from fat, carbohydrate, and proteins. So from these studies also, they revealed that all or a portion of the carbon skeleton of every amino acid is convertible either to carbohydrate. So there are 13 amino acids. So again, you have to memorize this. 13 amino acids that can be converted back to carbohydrates, one amino acid that can be converted to fat, and 5 of amino acid that can be converted both to fat and carbohydrate. So this is found in your book. So as you can see here, we have 1 sa fat, so leucine, 5 sa both, which can be converted both to carbohydrate and fat, and here we have 13 glycogenic amino acid that can be converted back to carbohydrate. Okay, and so looking at this figure, so we can see that from here, from this table, so these are now the amino acids. So this one becoming again pyruvate, acetyl-CoA, then enters again the citric cycle or the citric acid cycle. Here, glutamate, all of this is broken down to glutamate, again becomes alpha-ketoglutarate. And also this one, to succinyl-CoA, to fumarate, asparagine to aspartate, back to oxaloacetate. So, so from protein broken down to individual amino acids, and the individual amino acid again is broken down and again enters the citric acid cycle. So of course, you also have to take note of this and memorize this kasi if you know this pinaka-basic, it will be very easy for you to take note of the individual catabolism of this amino acid. Basta you know lang at what point sila nako-convert into a certain metabolite or a certain intermediate, and at what point they enter the citric acid cycle. So if you can recall from our very first lecture in biosynthesis, di ba, it's the other way around, like alpha ketoglutarate, it's the opposite, going to glutamate and going to this way. So ito naman siya pabalik because it's the catabolism or the breakdown. So as I have said, if you have memorized yung very first lecture natin, this will be easy for you to recall na lang or to remember kasi it's just the opposite of that one. Okay, so we know that the very first catabolic reaction, as I have said, of most protein amino acid is the removal of the alpha amino nitrogen by transamination, diba? So we have already learned this, na pinakauna is removal of the nitrogen group, and in the form of ammonia, they enter the urea cycle para get rid by our body. So masama siya sa urine. So except yun na. So these are the three exceptions, yung proline, hydroxyproline, threonine, and glycine, because for this amino acids, there are alpha amino group do not participate in transamination. So they have another way of another process in their catabolism. So they do not begin with the transamination or the removal of the alpha amino nitrogen. So, and after that, after removal of the alpha amino nitrogen, so now the hydrocarbon skeleton that remains are then degraded to amphibolic intermediates. And this is now is what we are interested for this lecture. Kasi tapos na tayo sa alpha amino nitrogen na topic. Okay. So again, let's start with the following amino acids. So maybe familiar na kayo with this reaction kasi baliktad lang talaga siya from the biosynthesis. So here, asparagine and aspartate again form or can form back oxaloacetate, diba? So the all four carbons of asparagine and aspartate for oxaloacetate via reaction catalyzed by una asparaginase para siya maging aspartate, and from aspartate transaminase naman, and this now becomes oxaloacetate. So metabolic defects which involves this transaminase is actually incompatible with life. So maybe that's why there is no known case na there is a defect here in transaminase kasi it's incompatible with life. Ayun. So yun lang, 2 steps lang from asparagine back to oxaloacetate. Again, ito naman, glutamine and glutamate. So you already know this. Glutamine, again, catalyzed by glutaminase, so becomes glutamate. And then the glutamate also is acted upon by transaminase and back to alpha ketoglutarate. So enter again the tricarboxylic acid cycle. So proline, yung proline, so catabolism of proline takes place in the mitochondria. So proline does not participate in transamination, but this is one of the amino acids that do not participate in transamination. So its alpha nitrogen is actually retained throughout a 2-stage oxidation to glutamate. So proline is broken down to glutamate, and further on glutamate is broken down to alpha ketoglutarate as discussed in the previous slide. So it actually starts here with proline. And this proline is catalyzed by proline dehydrogenase, and it becomes this one, pyroline carboxylate. And subsequent oxidation to glutamate here na is catalyzed by another enzyme, pyroline carboxylate dehydrogenase, or also called glutamate semialdehyde dehydrogenase. So if you can see the end product here, so there are actually 2 important reactions here. So you have to take note of this kasi for the metabolic disorders that affects this enzyme here, so they have an effect on the body or in the human body. So ayun, and this one. So proline ends as initially glutamate, and again the glutamate, of course, we know undergoes transamination to become back to alpha glutarate. So ayun, so as I have mentioned, there are two metabolic disorders of proline catabolism. So they are just actually autosomal recessive traits, so they are still consistent with a normal adult life. So there is type 1 hyperprolinemia, wherein the metabolic block is at proline dehydrogenase. So metabolic block is at proline dehydrogenase here, so type 1. And here, no associated impairment of hydroxyproline catabolism. So they do not affect, for type 1, they do not affect the catabolism of hydroxyproline. For type 2, however, the metabolic block is at the next step. So here na siya, metabolic block. So type 2 and the second reaction. So it affects the second enzyme. And ayun, so since this enzyme is also participates in the catabolism of arginine, ornithine, and hydroxyproline, and since it has an effect in the catabolism of hydroxyproline, so both pyroline and this one, pyroline hydroxycarboxylate, are excreted. So that is its difference to the type 1, kasi sa type 1, it has no effect on the hydroxyproline catabolism. So for type 2, affected din ang hydroxyproline. Ayun. So you have to memorize kung at what point or at what reaction siya may effect or may metabolic block. Okay? So next amino acid is the arginine and ornithine. So the initial reactions in arginine catabolism is the conversion to ornithine. And ornithine, this is followed by transamination to glutamate semi-aldehyde. And syempre, pag mag-abot na ang glutamate, if we already have glutamate, ang next step na dyan is transamination and again back to alpha ketoglutarate. So arginine becoming ornithine and then glutamate and alpha ketoglutarate. So ano ang importance ng ornithine? If we can recall, di ba? If here muna. So mutations in the ornithine aminotransferase, or also called ornithine transaminase, elevate plasma and urinary ornithine, and it is associated with gyrate atrophy of the choroid and retina. So treatment for this, usually pag mga amino acid naman, diet restriction lang. So restricting dietary arginine. So there is a condition known hyperornithinemia, hyperammonemia syndrome. So if you can recall our urea cycle, di ba? So the ornithine from yung number 6 na reaction, if you can recall na there is already the formation of the urea plus ornithine, di ba? So the ornithine from the cytoplasm must go back inside the mitochondria. So if there is a defective mitochondrial ornithine citrulline antiporter, so there is an impaired transport of ornithine back into mitochondria. So what happens is there will be no of citrulline yung lalabas na there will be a problem in the urea synthesis. So ayun, yung function ng ornithine. So next amino acid is histidine. So histidine, catabolism of histidine proceeds via here. So from histidine, there is the 4-imidazolone propionate, and then the formation of FIGLO or N-formimino-glutamate. And from this naman, yung formimino group transfers, yung group, formimino group is transferred to a tetrahydrofolate. So ang end product niya is again glutamate and eventually alpha ketoglutarate. So histidine, here imidazolone propionate, and figlo. Tapos, eventually, there is a need for a tetrahydrofolate here para maging glutamate siya and further on to become alpha-ketoglutarate. So, ayun, may mga blocks din dito which you have to take note. So, here, and also if there is a problem in the folic acid or in the folate. So, wait lang. Histidine. So, ayun. Ayun. So, as I have mentioned, important ang folic acid kasi kailangan siya sa transfer of the 4-amino group. If this is impaired and therefore, mag-i-stop siya here. So, figlo ang may excrete. Okay, so it cannot further on be converted to glutamate and alpha-ketoglutarate. Ayun. So, and that happens if there is folic acid deficiency. So, excretion of figlo following a dose of histidine can actually be used to detect if a person has a folic acid deficiency kasi hindi na na-break down ang figlo because it needs folic acid. So benign disorders of histidine catabolism include histidinemia and urocanic aciduria associated with impaired histidase. So here, so if there is an impaired histidase, mangyayari is, ayun, histidinemia and urocanic aciduria. Okay. So after histidine, we go to glycine. So ang glycine, there is a glycine cleavage complex of liver mitochondria which splits the glycine to CO2, ammonia, and forms methylene tetrahydrofolate. So ito ang reaction niya, yung glycine. And the glycine, there is a glycine cleavage system which consists of actually 3 enzymes. So 1, 2, 3, and an H protein that has equivalently attached dihydrolipoyl moiety. So this is how the glycine is catabolized. So there are conditions like non-ketotic hyperglycemia. This is a rare inborn error of glycine degradation and it is known in Finland. So here the glycine accumulates so there's also primary hyperoxaluria. So there is a failure to catabolize the glyoxylate formed by the deamination of glycine. So after glycine, after deamination becomes glyoxylate. So lithiasis, nephrocalcinosis, and early mortality from renal failure or hypertension. Another condition associated with an impaired glycine metabolism is glycinuria, and this results from a defect in renal tubular reabsorption. Ayun. So ito sila these three are conditions that are affected in if there is problem in glycine metabolism or catabolism. Okay, so next is serine. So for serine naman, initially it is converted to glycine. So converted to glycine by an hydroxymethyltransferase, by glycine hydroxymethyltransferase. And after it becomes glycine, it now proceeds yung allied na glycine catabolism. So broken down siya to glycine. So, ang alanine, we already know that yung transamination, so alanine is like glutamine, so they participate in transamination. So, after transamination, they form pyruvate. So, there is also no known metabolic defect of alanine catabolism. Kasi transamination lang siya and it is said that there is no, if there is a problem in transaminase, it is incompatible with life. So that's why we will never know if there is a defect that affects a transaminase. Ayun. So next amino acid is cysteine and cysteine. So here, from cysteine, it is first reduced to cysteine by a cysteine reductase. So L-cysteine. And after which, it follows two different pathways that convert cysteine to pyruvate. So here, yung top pathway is called the cysteine sulfinate pathway. So this one on top. Well, the one here, lower figure is the 3-mercaptopyruvate pathway. So they have actually two pathways for cysteine for it to become pyruvate as the end product. So there are defects here, the cysteine lysinuria or the cystinuria. There is defect in renal reabsorption of cysteine, lysine, arginine, and ornithine. And here, apart from the cysteine calculi, actually cystinuria is benign, but they can form mga cysteine calculi. So here, the mixed disulfide of L-cysteine and L-homocysteine excreted by cysteinuric patient is more soluble than cysteine and reduces formation of cysteine calculi. So this is the structure of the mixed disulfide of cysteine. So this is actually more soluble than cysteine lang. So by forming this one, there is a reduction of formation of cysteine calculi. So problems in the catabolism of cysteine and cysteine. So we have the vitamin 6 responsive and the vitamin B6 unresponsive homocystinuria. So here the deficiency in the reaction catalyzed by glutathione synthase. So there is a deficiency in the reaction catalyzed with this enzyme, and the consequence is osteoporosis and mental retardation. So another one is the cystinosis or the cystine storage disease, wherein there is a defective care-mediated transport of cysteine. So what happens, there's a deposition of cysteine crystals in tissues, and this can lead to early mortality from acute renal failure. So, those two are the problems that we may encounter if there is a defect in cysteine and cysteine catabolism. So moving on, we go to threonine. So threonine is cleaved to glycine and acetaldehyde. So glycine and acetaldehyde. So itong glycine, di ba, we can recall from previous slides, so they have its own catabolism na. So yung acetaldehyde naman undergoes oxidation to become acetate here with formation of acetyl-CoA. Ayun. So, threonine, first divided to glycine. Glycine undergoes its own catabolism, acetaldehyde, eventually forming acetyl-CoA. And, wait lang. So, ayun. So, for this one naman, so this is the catabolism of 4-hydroxyproline. So, yung hydroxyproline, catabolism of hydroxyproline forms pyroline hydroxycarboxylate. Ito siya, pyroline hydroxycarboxylate. Then hydroxyglutamate, semi-aldehyde, erythrohydroxyl glutamate, and alpha-ketohydroxyglutarate, which eventually becomes glyoxylate and pyruvate. So here, you also have to take note of the mga blocks here. So 1 and 2. So yung mga important enzymes involved here. So here, an aldyl type. So from this one, this ketohydroxyglutarate, there is an aldyl type cleavage which eventually forms glyoxylate plus pyruvate. So there is a defect here which is called the hyperprolinemia, and there is a defect in 4-hydroxyproline dehydrogenase. So here. This is just benign, so defect in the 4-hydroxyproline dehydrogenase. So there is no associated impairment of proline catabolism, and defect in glutamate semi-aldehyde dehydrogenase is accompanied by excretion of pyrroline hydroxy 5-carboxylate. Kasi, so if there is a defect here sa enzyme, so magbi-build up ang previous metabolite. So what happens is, ayun, ang may excrete is the pyrroline hydroxy carboxylate. So, next amino acid is tyrosine. So, yung tyrosine naman starts with transamination. So, tyrosine starts with transamination to hydroxyphenyl pyruvate with successive hydroxyphenyl pyruvate with successive formation of mallyl acetoacetate, fumaryl acetoacetate, fumarate, acetoacetate, and ultimately acetyl-CoA and acetate. So here, medyo marami ang defect for tyrosine. So 1, 2, 3, 4. So 4 enzymes can have a defect or can be affected in defects for tyrosine catabolism. So, ayun. So, we have this. We have the type 1 tyrosinemia or tyrosinosis. So, there is a defect of the enzyme fumaryl acetoacetate hydrolase. So, therapy employs a diet low in tyrosine and phenylalanine. Untreated acute and chronic tyrosinosis can lead to death from liver failure. Then we also have a type 2 tyrosinemia or the Rickner-Hanhart syndrome. So the defect is the tyrosine aminotransferase. So since there is a defect in this enzyme, alternate metabolites of tyrosine are excreted kasi yun yung nag-build up. The catabolism cannot progress anymore to form the end product. Then we also have the neonatal tyrosinemia. So this is due to lowered activity of p-hydroxyphenyl pyruvate hydroxylase. And again, therapy is just diet modification. So diet low in protein. We also have the Acaptonuria. So, ang Acaptonuria is actually yung pinaka-common or usually yung naririnig natin if we talk about defect in tyrosine catabolism. So, here there is a defective homogentisate oxidase. So, what happens in Acaptonuria is the urine darkens on exposure to air due to oxidation of excreted homogentisate. So, there is arthritis and connective tissue pigmentation in an individual because of the oxidation of homogentisate to benzoquinone acetate, and this polymerizes and binds to connective tissue. So this is yung pinaka-familiar or yung disease actually na na-attribute in defect in tyrosine catabolism. So you will often be hearing about Acaptonuria. So from tyrosine, we go to phenylalanine. So phenylalanine is first converted to tyrosine. So from phenylalanine, it becomes tyrosine. So as you can see sa previous slide, it then follows the tyrosine catabolism. So there are also problems in phenylalanine catabolism, and this becomes hyperphenylalanemia. So we have the type 1, type 2, 3, type 4, and type 5. So if you can see the type 1, these types depends on what type of enzyme that is affected. So for type 1, the defect is that in the phenylalanine hydroxylase. So type 2 and 3, the defect is in dihydrobiopterin reductase. Type 4 and type 5, the defect is in the dihydrobiopterin biosynthesis. So, ayun. So if there are defects, alternative catabolites will be excreted. And treatment should be a diet low in phenylalanine so that this can prevent actually mental retardation. Ayun. So yun lang, diet modification also is important. Okay, so after phenylalanine, we have the lysine. So yung lysine, first reactions of lysine catabolism in the human liver form protonil-CoA, and this protonil-CoA is degraded to acetyl-CoA by the reaction of fatty acid metabolism. Ayun. So here, yun. So there are also to form protenyl CoA from lysine, there are actually 6 reactions. So reaction 1 and 2 convert the shift base formed between alpha ketoglutarate and the amino group of lysine to amino adipate semialdehyde. So kasi if we can recall from the yung biosynthesis, nag-form siya ng shift base. There is actually a formation of shift base between alpha ketoglutarate and lysine. So, and the amino group of lysine. So yun yung initial reaction naman sa catabolism. So there is na-convert yung shift base to to amino adipate semialdehyde. And so this both reaction, the 1 and 2, are catabolized by a single bifunctional enzyme. This one, amino adipate semialdehyde synthase. Ayun. Tapos, after that, there is the reduction of the amino adipate semialdehyde. So there is now, this is followed by transamination to become alpha ketoadipate. Ayun, so here, transamination with the use of alpha ketoglutarate, and there is the formation of glutamine. So here, there is from amino adipate, ketoadipate is formed. So conversion to the thioesters glutaryl-CoA here is followed by decarboxylation of glutaryl-CoA to crotonyl-CoA. And from this one, subsequent reactions are sa fatty acid catabolism na. So as you can see, yung lysine participates sa fatty acid catabolism. So problems in lysine catabolism can result to hyperlysemia. So hyperlysemia, this results from a metabolic defect in either the first or second activity of the bifunctional enzyme amino adipate semialdehyde synthase. So here, reaction 1 and reaction 2. So this is accompanied by elevated levels of blood saccharopein. If the defect involves the second activity, so there is an accompaniment or it is accompanied by an increased level of blood saccharopein. Kung yung involved na metabolic defect is sa second activity. So metabolic defects at reaction 6 naman, so reaction 6, so here, results in an inherited metabolic disease that is associated with striatal and cortical degeneration. So what happens is there is elevated concentrations of glutarate and its metabolites, glutaconate and 3-hydroxyglutarate. Kasi it cannot further on proceed sa catabolism. So there is a build-up of the metabolites prior to the defect in the reaction 6. So the challenge in the management of the metabolic defects in lysine catabolism is how to restrict the diet or the entire intake of lysine without producing malnutrition. Kasi as you can see, diba, they are involved sa fatty acid metabolism. So ma-affect ang metabolism also if you do not have lysine. So yun ang challenge actually ng treatment for lysine, problems in lysine catabolism. So moving on, we have tryptophan, which is another amino acid. So the initial reaction is the degradation to amphibolic intermediates via the kynurenine antranilate pathway. So, ayun. tryptophan 2,3-dioxygenase opens the indole ring and incorporates molecular oxygen and forms, so dito, so what dito siya. So this opens the indole ring, this enzyme, tryptophan pyrrolase, and with the use of or incorporates molecular oxygen and forms this one, N-formylkynurenine. And hydrolytic removal of the N-formyl of the formyl group. So dito, hydrolytic, so with the use of H2O of the N-formylkynurenine catalyzed by kynurenine formylase produces this one, kynurenine. Ayun. So oxidation and then hydrolytic removal of the formyl group. So since kynurenase requires pyridoxal phosphate excretion of xanthorrhinate in response to a tryptophan load, so if there is a build up of this one, so it is actually a diagnostic of vitamin B6 deficiency kasi yung kynurenase requires pyridoxal phosphate or vitamin B6 in order for it to form xanthorrhinate. So if there is no vitamin B6, if there is excretion of xanthorrhinate, meaning there is a deficiency in vitamin B6 kasi hindi nakaka-proceed ang reaction or yung catabolism further down the reaction. Okay, so ang pinaka relatable disease for tryptophan if kanina is phenyl is the heart-nap disease. So if we here heart-nap disease, so it's usually related to a tryptophan catabolism defect. So here it reflects impaired intestinal and renal transport of tryptophan and other neutral amino acids. So the indole derivatives of unabsorbed tryptophan formed by intestinal bacteria are excreted. So the defects limits tryptophan availability for niacin biosynthesis, and this one accounts for the pellagra-like signs and symptoms. So you will be hearing this na pellagra-like signs and symptoms. So heart-nap disease as a consequence of a problem in tryptophan catabolism. Then we have the amino acid methionine, which first reacts with ATP forming S-adenosyl methionine. So this is an active methionine. So there is a requirement for an ATP in its initial pathway in catabolism. Then subsequent reactions form propionyl CoA, which then subsequently is converted to succinyl CoA. Ayun, so propionyl CoA. And yung succinyl CoA, if we can recall, diba, goes back sa tricarboxylic acid cycle. So it enters the cycle again. Okay. So now we go to the 3-branch chain amino acids. So here we are talking about isoleucine, leucine, and valine. So the initial reactions are common, actually common to all these 3-branch amino acid, and their reaction are analogous to reactions of fatty acid catabolism. So here, same sila lahat, and ending up with this one, formation of methylprotonyl CoA, methacrylyl CoA, and diglyl CoA. So and after that, so following transamination of the carbon skeleton, so actually here, it starts with here, sa transamination of the alpha keto acid. So they following this one, the carbon skeletons of the resulting alpha keto acid undergo oxidative decarboxylation. So here, and conversion to coenzyme A thioesters. So this one. So it starts with transamination, then followed by oxidative decarboxylation to form this coenzyme A thioesters. So this multi-step process is catalyzed by the mitochondrial branch chain alpha keto acid dehydrogenase complex. So ito siya is actually catalyzed by the alpha keto acid dehydrogenase complex, and it has 5 components. So it has E1. So this is the thiamine pyrophosphate dependent branch chain alpha keto acid decarboxylase. So these are the enzymes involved in this complex in order for this 3 branch chain amino acid be converted into this coenzyme A. So we have first E1, thiamine pyrophosphate dependent branch chain alpha keto acid decarboxylase. Then we have E2, dihydrolipoyl transacylase. So this contains a lipoamide. Then we have enzyme 3, dihydrolipoamide dehydrogenase, which contains FAD. Then we have protein kinase and protein phosphatase. So all these 5 is the component of this alpha keto acid dehydrogenase complex. So the protein kinase and protein phosphatase regulate activity of the branch chain alpha keto acid dehydrogenase complex via phosphorylation and dephosphorylation. Ayun. And dehydrogenation of the resulting coenzyme A thioesters proceeds like the dehydrogenation of lipid-derived fatty acyl-CoA thioesters. So, 5 enzymes are involved sa pinakaunang reaction to form this coenzyme A thioesters. And this now proceeds like the dehydrogenation of lipid-derived fatty acyl-CoA thioesters. So, ayun, these are the individual reactions na for the further breakdown of the individual branch chain amino acid. So, this one is for leucine, this one is for isoleucine, and this one is for the valine. So, there are 3 amino acids that undergoes this type of catabolism. Okay, so ayun, konti na lang. So metabolic disorders of branch chain. So we have the following metabolic disorders. Earlier, di ba, for each amino acid, we tackle about the different metabolic disorders in relation to a particular amino acid. Now for this one, for the branch chain, so ito naman ang metabolic disorders nila. So we have first the maple syrup urine disease. So, or the branch chain Catabolism or the MSUD. So here, what happens is the odor of the urine suggests maple syrup or burnt sugar. So, if you will hear a maple syrup urine disease, so it is a disorder of the branch chain amino acid catabolism. So, the biochemical defect involves the alpha keto acid decarboxylase complex. So, di ba yung complex may 5 enzymes? So, yun sila. There is a defect in that area. So, what happens is plasma and urinary level of leucine, isoleucine, valine, and their alpha keto acid and hydroxy acids are elevated. But here, the urinary keto acids, yung nakikita sa urine, is mostly contributed by leucine. Ayun. So, since may problems sa enzymes, so this complex, ayun. So, there is the build-up of this amino acid and their intermediates. So signs and symptoms of MSUD includes fatal ketoacidosis, neurological derangement, mental retardation, and of course the maple syrup odor of the urine. So the mechanism of toxicity is still unknown, but early diagnosis of enzymatic and by enzymatic analysis is essential to avoid brain damage and early mortality by replacing dietary protein by an amino acid mixture that lacks isoleucine, leucine, and valine. So dietary modification again. So since there is a problem in breaking down this branch chain amino acid, so by replacing them, this can actually help in avoiding brain damage and mortality. So the result comes from the mutation in the genes that includes the alpha, the enzyme 1 alpha, enzyme 1 beta, enzyme 2, enzyme 3, diba? So earlier we already mentioned this, what are these enzymes? So there is a type 1A MSUD, type 1B MSUD, type 2, and type 3. And these types depends on what enzyme is affected. So E1 alpha, E1 beta, E2, and E3 genes are affected in these different types. And this one is the table that shows the enzymes affected. So here, diba, enzyme 1, so the alpha ketoacid decarboxylase, enzyme 2 dihydrolipotransacylase, and enzyme 3 dihydrolipoamide dehydrogenase. So any defect in this enzyme can result in the different types of maple syrup urine disease or MSUD. So we also have this, the intermittent branch chain ketonuria. Here, the alpha keto acid decarboxylase retains some activity. So, ito siya intermittent lang. So, may activity pa rin siya na unlike the MSUD na there is really a defect. Ito siya, they still retain some of its activity. So, the symptoms occur later in life. Then we also have isovaleric acidemia. So, what happens is the ingestion of protein-rich food elevates isovalerate. So there is an impaired, the impaired enzyme here is isovaleryl-CoA dehydrogenase, and this results to vomiting, acidosis, and coma. So those are the presentation of the patient. Okay, so I think yan. So summary na pala. Okay. So, ayun. So, we have no choice pero kailangan natin i-memorize yung individual enzymes involved in the catabolism of the carbon skeleton, especially for those enzymes na with diseases yung we usually hear yung phenyl ketonuria, yung maple syrup, yung MSUD. So yun kasi they usually come out kahit not in biochemistry, even in other subjects kasi yun sila yung pinaka-common diseases associated with a certain amino acid problem in a certain amino acid catabolism. So again, excess amino acid, di ba? We have already learned this na yung amino acids, yung protein in our body is broken down in its component amino acid, and the excess amino acid are catabolized back to amphibolic intermediates that serve as a source of energy or for the biosynthesis of carbohydrates and lipids. So, di ba, yung mga end product here, we can recall na they go back as alpha ketoglutarate needed sa carbohydrate synthesis, or as succinyl-CoA, or as pyruvate, or acetyl-CoA. So, ayun. So, from that, we can see na interconvertible talaga from protein, carbohydrate, and lipid. So, transfer lang, and they just form another metabolites or another intermediates. Ayun. So transamination is the most common initial reaction. So we already know this, diba? So except for, so there are exceptions to this, yung hindi nagpa-participate sa transamination. So you have to know those 4 amino acid that do not or do not participate in transamination. But for all the rest, transamination muna is the first or the most common initial reaction of amino acid catabolism. And subsequently, subsequent reactions remove any additional nitrogen and restructure hydrocarbon skeletons. So yung natitira, yung carbon skeleton is converted back to acetyl oxaloacetate, alpha-ketoglutarate, pyruvate, and acetyl-CoA. So metabolic diseases associated with glycine catabolism, glycinuria, and primary hyperoxaluria. So metabolic diseases yan associated with glycine. So there are 2 distinct pathways that convert cysteine to pyruvate, diba? So dalawa yon. I have shown the picture. So may top pathway and yung nasa baba ng pathway. So metabolic disorders of cysteine catabolism include cysteine lysinuria, cysteine storage diseases, and the homocysteinurias. Threonine catabolism merges with that of glycine after threonine aldolase leaves threonine to glycine and acetaldehyde. So, ayun, yung threonine, di ba? So, we have already shown this na break down muna or broken down to glycine, and the glycine goes its own catabolism to acetaldehyde, which also progresses to another pathway. Following transamination, the carbon skeleton of tyrosine is degraded to fumarate and acetoacetate. So metabolic diseases of tyrosine catabolism includes tyrosinosis, Rickner-Hanhart syndrome. So may mga type 1 tyrosine, di ba type 1, there are different types of tyrosine catabolism problems. So then we have neonatal tyrosinemia and Acaptonuria. So this all depends on what level or enzyme block is affected. So metabolic disorder of phenylalanine catabolism includes phenylketonuria, phenylalanine, and several hyperphenylalaninias. Then neither nitrogen of lysine participates in transamination. So the same net effect is however achieved by the intermediate formation of saccharopene. Metabolic diseases of lysine catabolism include periodic and persistent forms of hyperlysinemia and ammonemia. Okay, so ito siya, these are the branch chain amino acids. So the catabolism of leucine, valine, and isoleucine presents many analogies to fatty acid catabolism. So metabolic disorders of branch chain amino acid catabolism includes hypervalinemia, so yung maple syrup urine disease, intermittent branch chain ketonuria, isovaleric acidemia, and methylmalonic aciduria. Okay, so yun na pala ang last. So, any questions?